everyone. So today we are welcoming Joanne Benett, which is from Charles Sturt University, and we will talk to you about conserving species.
Yeah.
I will let you tell the result, it will be easier. And thank you everyone for being here. And so I will let you begin.
Yeah, that's not going to work. Put it on the floor. Hi. So I'm really going to talk today about a collection of work that's from a big fellowship that is about to finish this year, and a big title because I wasn't sure how else to stick it all together. Just to say a little bit about myself, sort of my research has probably been a bit varied, but a big component of it is looking at how climate and land use have been interacting to affect species distributions and the interactions between species, and then the outcomes for ecosystem services and function. And so I have worked a lot in even the thermal physiology of species and climate adaptation, but also I actually even started on woodland birds and large-scale species distributions at landscape scale and things like that. So, and it all fits together, I promise. But today I'm mostly going to be talking about my current work that's on pollination. I think I'm probably not saying anything that nobody in the room already knows, but I sort of think personally, pollination is probably one of the key regulators of plant populations. About 90% of the world's plants are meant to be pollinator dependent, and 75% of crops, and in reality that's probably an underestimate. And I feel like pollination plays a role in all of the things I really like coffee and chocolate. And I also think that's a big part of our society and cultural. I think if the world didn't have pollinators, we'd all be eating a lot of bread, and that would be really sad. And we've all become quite concerned about pollinator declines. There's pretty good evidence that we are losing insect species. But most of the information comes from Europe and America, and there's big areas of the world where we've got no information at all. And actually, where I'm from, Australia is one of them. The big IUCN global assessment, Australia is a big grey on the map, which is really disappointing because we're, a wealthy country and there's lots of research happening. But I'm one of a few people who apparently really dig bugs. And I think the only reason you could possibly have for not liking insects is you haven't looked at them for long enough. They do really cool things. And there's, pollinator declines have made people start to worry about a pollination crisis where plant reproduction fails. But there's actually, whether or not pollinator declines will lead to plant reproduction losses is a bit complicated because plants, they've got their own traits and their own problems. And there's whether or not a plant's reproduction is limited by receiving pollen, so pollen limitation, strongly depends on the plant's traits itself. And whether or not that will have an effect on plant populations in the long run depends on the plant's demographic strategies. So lots of plants can be highly pollen limited, but it doesn't actually matter for them because they're long-lived and they can spread the risk over their lifetime. And as long as they get pollinated once, they'll be able to replace themselves. So you'd think probably that the strongest responses of pollinator declines will be in short-lived species. But short-lived species have a high pressure to not be pollinator dependent if they have evolved in an environment that is variable for pollination. And there's various ways plants do this: autogamy, self-fertilization, and often this is on a scale of how deeply they self-pollinate. And then they, clonality, wind pollination. The other thing they do is they can be super generalist. So they interact with tons of pollinators. They don't really care who rocks up, like this ranunculus, it's just, all out there. And then, or they interact with super generalist bees, but they might have more complex floral structures, but they're still able to pollinate with something that's like highly abundant in the environment. This sort of, I think in the general public, certainly when I talk to my family members, they sort of think like over here, like Darwin's orchid, that like it's a one-to-one relationship most of the time, and that you're going to lose a pollinator, that means you're going to lose a plant. But that doesn't really happen, and that's partly because of this horrendous thing, which I've actually cut off because it just keeps going forever. But there's always like usually way more pollinators in an environment across the top than there are plants. And these plants, the ones that are only interacting with one species, which I think I've got an arrow that points to them, it's the most abundant pollinator in the system. So hopefully that introduces what I'm going to hopefully talk about today, which is, you know, how vulnerable really are plants to reductions in pollination? Are plants becoming more pollen limited through time? You know, what is driving these changes in interactions? And if you do observe a change in plant-animal interactions, like can that change be used to predict outcomes for plants, really? And is pollen limitation so bad in some plant populations that it can actually be causing population decline in those plants? So how do we look at changes through time in a country like Australia where we have no data on our pollinators? Even, I think it's like almost a half, I forgot what the statistics is now. It's nearly 2,000 species of bee aren't identified in Australia. And it's much, much worse for flies and things like that. So we don't know what we've got. We really have no idea. But the ways that we've been trying to do it through this fellowship is you're just resampling collections where we do have collections, and then doing sort of modern day field comparisons, and also, you know, collecting data from published studies and trying to synthesize that. What we've been, and part of this sort of synthesizing all this data is using experiments where people have, to answer this first question about how vulnerable are plants to pollination losses, is experiments where people have actually tried to figure out pollinator dependency, or sometimes it's called pollinator contribution. And it's a really simple experiment. People usually do it to describe a breeding system of a plant. And it's just, you compare a hand supplemented plant to a plant that is bagged, so the pollinators are excluded. And that tells you how much a pollinator has contributed to the reproduction of that plant. So, and this, like I said, it's usually done to describe a breeding system. It's been done thousands of times. I think we had, this is part of a broader data set called Global. It's Global Pollen Limitation, but as part of that, we also collected information from bagged plants. And so this is about 1,500 experiments, I think, up here, and almost all different species. I think nearly like 1,200, 1,300 plant species. So, you know, the first step is to really sort of understand how vulnerable are plants. And actually, the data is pretty well distributed across the tree of life. And we found that it's quite skewed. So these are the plants that produce no seed if they're bagged. So it's a lot of plants produce no seed if they're bagged. But you consider a plant to be pollinator dependent if it produces less than 20% of its total reproduction potential in a bag. And so about 80% of plants are pollinator dependent from this data set, and about half would produce no seed at all. And so then the next question we asked is, you know, if plants are super vulnerable to pollinator declines, are they experiencing reductions in pollen receipt, and if this is affecting their ability to produce seed? And this is another really simple experiment where people supplement by hand pollen to saturation, whatever that is. Usually you just put as much on as you possibly can. And then you compare it to one that is open to pollinators. And this is just over 2,000 species. And we wanted to ask what are the drivers of pollination service loss. And so I'm still missing connective tissue. We wanted to ask what are the global change drivers that are actually maybe driving declines in pollination service and outcomes for plants, which is the second question. And then we overlaid it with global distribution information on land use. And this is homogenized land use. And we put it into 3 broad categories. And managed is any agricultural sites. And this includes rangelands here, actually. But we took them out and put them back in and did all sorts of sensitivity analysis. It makes no difference, because rangelands are a bit whether or not they're natural. And then national parks and urban centres. So there's about 190 urban centres here where people have done these experiments in the middle of cities. And then the rest of the data is split between agricultural and natural sites, despite they're not, probably the vast majority of the globe is more managed. But I think that's reflective of the fact that people like doing field work in nice places. On a global scale, about 60% of plants are pollen limited, so it's super common. And again, it's well spread across the tree of life. You can see that pollen limitation is in orange, and it's very common. Plants are commonly pollen limited. I think the average effect size is about a 60% increase when supplemented compared to the open, naturally open. We split the plants based on the categories I showed before, pollinator dependence versus actually dependent on pollinators. Otherwise, pollen limitation doesn't necessarily make sense in a pollinator context because it could be wind. So everything, this dotted line here is autofertile or wind pollinated plants. And then the line above is the pollinator dependent. And so as you'd probably expect, things that aren't pollinated by insects are not pollen limited. And things that are pollinated by insects are. But it's the same in natural habitats compared to managed, using these very broad categories. But it's much higher in urban centres. We also really wanted to look at the relationship with pollinator groups. We were really only able to use sort of broad categories because we really don't know what the most important pollinators for most plants are, or it's, they're visited by so many things. But, and we only did this for the natural and managed sites because although the urban centres is about, nearly 200 different cities, there's not enough data within that to start splitting it apart. So we looked at ecological specialisation. It's not that common, but if you really are only pollinated by one taxonomic group, you are much more pollen limited than anything else. So this is this idea of sort of ecological generalisation is sort of protects you from pollination change. We tried to split this into species that have or are thought to have specialised relationship on different taxonomic groups. So we had, and again, the data set's not large enough to look at things like flies and butterflies. So exclusively other is everything that is not pollinated by a bee. And we found this weird relationship, but I'm not sure that we thought was probably to do with honeybee provisioning. But in natural habitats, if you have a strong association with bee pollination, you're much more pollen limited. But that doesn't hold in managed habitats. And so it seems like land use has this really strong effect on pollen limitation. And one of the like early works on this was this study here by Sol Cunningham, where he, I think in the 2000s, went out to the mallee woodlands of Australia and did these experiments, did pollen limitation experiments on about, I think it's 16 field sites. And he showed that increasing fragmentation, more isolated sites, smaller sites, the plants are way more pollen limited. And he did this just focusing on these four species here. This species is really cool because it's buzz pollinated, which is unusual for Australia because we don't have any bumblebees. And so it has a very specialized bee relationship because we don't have that many bees that are capable of buzz pollination. And then we have these classic acacia with the big yellow orbs, and they can basically be visited by anything. And then this Eremophila species. This senna species is a bit weird because it requires pollinators to remove its anther caps, but it's self-compatible, so it's weird. But even that one was more pollen limited, so it's for some reason receiving less visits, even though it then self-fertilizes. So this is where the region is, the middle of nowhere in Australia officially. And this is what it looks like. So these are huge agricultural fields. You know, these are tiny fragments here. And then all the way up to this nature reserve up the top, that is the size of a small country. It goes for miles and miles all the way into the Red Desert. This is what the sites look like. I love them. They're dominated by about, actually, probably 5 different shrub species. There's not that much understory. They're quite homogeneous because they are extremely resource limited, both soil nutrients and water. So they're pretty similar, not that diverse for the plants, but they're really cool. This is super preliminary data. I just really wanted to talk about what we're doing. For all the shrubs, we've redone the pollen limitation experiments. In the first year, it was really depressing because you can imagine how hard it is to get out there. And we're out there with a group of students, and we did all the experiments, and we came back 2 weeks later, and there'd been a heat wave, and all the plants are SNEST. So it's just, and it's a bit like this week here, actually. We were there, and it's in the 30s, which is normal temperature, but we had a heat wave in October, which is spring, of in the 40s, and it's a major event that's extremely unusual. It gets hot there, but it shouldn't be that hot at that time. So we lost all our plants, and then you can't even go there because of the fire risk. So you can't, it's not as if we could have even gone and saved them. It's done. We also did, so we redid Sol's experiments. But what Sol didn't do is he didn't really he didn't have any information on the driver beyond fragmentation. So he collected information on the soil from the sites and found that it's basically the same. And it's all water limited, pretty homogeneous. So presumably it's something that's driven from the interactions with pollinator species. But he didn't collect visitation data. So we've gone back and we've collected visitation data. We've measured the insects for their body size. We've taken pollen from their bodies. We've collected stigmas for pollen deposition. And we've also done breeding system experiments on the plants. My student, Alana, focused on the acacia. And so it's the one part of the data that is finished. And she found some pretty strong results. This is, I stole straight from her honours thesis. I just copied and pasted it. On the fragments, so these, we split the small fragments and compared them to the large reserves, the reference sites. For some reason, she doesn't find any small bees on the fragments, which makes sense because it's probably about dispersibility. We know small bees can't disperse very well. There's plenty of studies showing that there's this size effect where you don't find small bees in more isolated sites. But she doesn't even find large bees on the reference sites, which I find a bit weird, but she didn't find any. There seems to be a big effect on the bee body size, so maybe they're carrying different pollen, and she painstakingly took all the pollen off all the bees and identified the plant species, and the pollen composition on the bees is exactly the same. And the proportion of heterospecific pollen compared to concentric pollen is also exactly the same. So it's probably something to do with not so much about what they visit, but how they then deposit pollen on the stigmas of the plants. Because this plant is still more pollen limited. Well, the year it worked, it's still more pollen limited in the fragments. And again, this is sort of the same sort of idea. I love this project. This is another one of my students, Okarite. We were contacted by the local Canberra government in Australia, and they wanted to know how to manage their grasslands. So Canberra, as a city, is full of these endangered grasslands, slightly different habitat types, which is what the map shows. I think there is like grassland, and then there's grassland, open woodland, and grassland, shrubland. And they wanted to know, how to manage them. Traditionally, they would have been burnt, and that's how they're managed. But now they're not. And so they don't, in the 70s, they decided to protect them, and the decision was basically to lock them up. But the problem is over time, they've become grassy, where before they would have been full of flowering plant species, but fire would have controlled the biomass. So they want to know what they should be planting, what seed mixes they should be throwing around, and how they should be controlling the biomass. And so the first question was like, how do we try and figure out what even was flowering there in the past? Canberra is a city that was chosen, its location is chosen, because Australia is a federation of states, and the power is actually at the state level. The federation is about cooperation between the states. So Canberra was chosen because Melbourne and Sydney couldn't decide who would be the capital. And it's almost still an ongoing battle of who should be the capital. So they chose Canberra. But as part of the committee of people who were going, they had a few different places they were considering. A guy who was part of this, I think he was an engineer, he collected bees. So in what is now Canberra's Parliament's lawn, and it's all landscaped, we have a really cool bee collection. And we also have a bee collection from down here, which is now, these grasslands are through the airport. And so Okarite swabbed all those bees in the collection and created a plant list for what used to be in these habitats. And then she's gone through, so it's a small collection, just from 2 places. But she's gone across all these different sites to try and catch as many bees as she possibly can from the same collection, so that she can swab them, the modern day collection, and do metabarcoding of the body pollen. And this is what Canberra's grassland looks like now. This is Blue Devil grassland, it's one of her sites. And these are all just through the city, and some of them are tiny. There's even one in the very center of the city that's like the size of this panel. It's still protected, it's tiny. And then some of them are quite large, like the big one that's in the north. And they've started controlling the biomass. And one of the ways they've done this is they let cows in, which doesn't work at all because Australian plants are, have evolved in depleted soils. We have very depleted soils in Australia. And so by having cows, they've enriched the soils, and then they had invasive species problem. So they're now taking the cows out, and they've started mowing, and they want to put some plants back. So unfortunately, in the old collection, there is a lot of these collected bees. About 50% of Australian bees are from the collected family. They have Gondwanaland evolution. They're thought to have co-evolved with Myrtaceae. We weren't really able to find those species, which probably reflects the fact that they are really specialist. Ocarite's data is kind of dominated by these species, which are all laser glossum. And they have really flexible behavior, but they don't fly very far. And they're really, like, most Australian bees are little things. We don't have, most of them are solitary. The European honeybee in the Mallee is the largest species that we find. So it's mostly small species. They're really cute. And we catch them and put them in pins. So this is, the black is what was shared between the past and now. The grey is historical only, and blue is today. They're interacting with so much stuff. And a lot of it, it's not just like invasive species, it's native species that are ornamental that people plant in their gardens. There's a lot of native species that don't belong in Canberra in this blue. There's cucumber, tomatoes, pumpkins, everything from your back garden. So basically so they now happen to interact with everything. There's a whole bunch of species that we don't find anymore in the environment. And there is ways that we can reintroduce them, because if they require fire to germinate, there are experiments at the National Seed Collection, which is also in Canberra, where we're like heating seeds up and exposing them to smoke. And so eventually it is the hope that we can go around with packets of priorly burnt seeds and make a difference. So I've really focused a lot on like the effects of land use on pollinators and like pollen limitation, which is a big focus of my work. But something we really would have loved to have done with the pollen limitation stuff is to look at whether or not climate change is a major driver of change. No one's ever done pollen limitation through time. Oh, sorry, that's a lie. There's one study, it's from America. They found pollen limitation increased through time, and then they did the experiment for a few more years and the effect went away. So there's one long-term data set. It's in the data, and that's it. And so then we thought we'd use climate velocity. So this idea, like maybe if we switch space for time and environments that have had big increases in climate, we can compare those that haven't had such big increases in climate. But just like, I think every big synthesis, almost all our data comes from North America and Europe, and so the climate is, the deviation in the climate velocity is hopeless. It's like less than 0.1. It didn't work. So then we started to think about how do we look for changes in, plant reproduction as a consequence of changes in pollinator interactions for plants, when we just don't have the data and there is no, well, other than one study, there's no historical pollen limitation data. So we've gone back to our historical collections, and we've done this using both insect collections and herbarium data. So looking at changes in stigmatic pollen deposition through time, but also changes in interaction between plants and animals, and then trying to see if it's in any way predictive of changes in plant reproduction. So the first place I'm going to take you to is the Australian alpine. A lot of people think that Australia doesn't have an alpine. It does, it's just really small. That's our alpine region. It's real. There's snow on the top, it's summer. It's got one tree species, that's the tree line. And then we have the field sites from the trio line that go up. Oh, that's not meant to be there, that's okay. And the one thing the Australian alpine has, or it had, is David Inouye in the 1980s. He came on holidays to Australian alpine with his wife and kid. And he went sampling insects almost every day. He set up 22 sites from the tree line up to the top, and he camped up there with his kid. He apparently even got lost once with his kid and was really frightened his wife would kill him when they turned back up. And he's great because he sent me all his field notebooks, which are just so detailed. And so like historical resampling has been done a few times. This is not super historical, but it's 40 years in an environment that's had rapid increases in temperature. But the great thing about this data set is he also recorded the relative abundance of the flowering plants. So we don't just know what the plants visited, we know what plants were there and how abundant they were. And the other wonderful thing David Inouye did was he tapped, I don't even know what to call it, markers into the ground, and they're still there and you can find them. They weren't GPSed or anything. He had a notebook where he described the location of the markers, and some of the descriptions are fantastic. He's like, you walk off the path 5 paces, leave the path when you see a rock that looks like a wombat. And so we're like, does this rock look like a wombat? I don't know. But anyway. And then eventually we were like, we found the site and we were like, yeah, that rock does look like a wombat. It's true, he's not wrong. And so we resampled the same plots for the plant-pollinator interactions, recorded the plants again, but this time we did pollen limitation through the season with the view that the plants that have had the biggest change in interactions maybe will be more pollen limited, was the hypothesis, or, you know. And we have exactly the same plant community, all the same plants are there. Nothing David Inouye sampled, we weren't able to sample. And then, so this is my student, Josh, up the top. I asked him for that photo, he sent it to me, I'm not mean. But that's also really typical of the Australian alpine. The weather is so variable. You can be up there and it's beautiful sunshine, and then suddenly it's hailing and lightning, and you're trying to run down as quickly as possible. So when David Inouye did it, actually, it was quite a warm year. But when Josh went up there, it was a super warm year and then a slightly warm year. So one year was pretty similar to David Inouye, and one year was much, much hotter. So Josh went up there. Josh actually had twice the sampling. I think David Inouye only took a day off of his holiday every other day to visit the plots. Josh went every day for 2 years. And the main thing we found actually is there's a big difference in the bees. This is David Inouye's bees down here in the blue. He found way more species than us. We couldn't find about 50% of his bee species. And so even though pollinator, they're super variable, but this is all seasons, it's 4 months of sampling every day for 2 years. And yeah, 50%, 4 genera we cannot re-find. The beetles are sort of in the middle. The flies seem to be the same, the community seems to be the same. And the butterflies, you know, David Inouye didn't record very many. We still don't record very many. There was never very many to begin with. And we swabbed their body for pollen, and we've never found a pollen grain on them. So there probably not a lot's going on from that perspective. There's been a huge change in phenology, actually, much more than we ever imagined. So this is David Inouye's in green. So even in the year, which is the first year of that's the first year of sampling in orange, that was like similarly temperatured. The plants are flowering much earlier, but it's not a small amount earlier. On average, it's 2 to 4 weeks. Some of these plants are flowering like 45 days earlier, even in the year that was like similarly warm, which is quite a lot. And people ski in the Australian alpine, but it's probably to do with snow melt, because we're finding that, yeah, the snow, if it comes at all. So in many years, we've had failure of snow. And it's really weird because you go up there and there's no snow, and clearly there's meant to be, because we've got ski fields with the ski lifts and absolutely no snow underneath. And maybe I should have found this video a couple of years ago. One of Josh's field sampling years, there was such a small patch of snow, and that's all that there was, that the Australian alpine ski group went up there anyway and did a weird little conga line on it and just went around in circles, because that was all they got for snow that year. So the ski lifts and everything, it's quite, it's not very high. So you don't want to be an Australian in an Australian ski club. So the first question was sort of like, are the plants, you know, what are the plants doing with their interactions? So this is just 6 plant species here. In terms of the interactions with their pollinators in the past, so David and Nui is in black, all of the plants were much more specialized in the past, were visited by less species than they are now. So they're still getting visited, but by more varied things. And so the next question was, are they pollen limited or are they pollinator dependent? Because if they're not pollinator dependent, the pollen limitation that they might be experiencing will be resources. And they're all highly pollinator dependent. None of them produce any seed in the bag. This is only a subset of the data because we haven't finished doing the pollen limitation for all of it. Yeah, and none of them are pollen limited despite these huge changes. And we've done the same thing in other places. In the Arctic, this is Liana Zola and Vivian Cox. She did her master's on pollen limitation in the Arctic, and Vivian resampled this guy's data set. So his name's Frans Sillen. He was a curator, insect curator at the Finland Museum. So his whole collection is at the Finland Museum. He kept really detailed notebooks, but this wasn't part of his job. For many years, he went on holiday to this very specific place, and he recorded all the insects he saw visiting plants for his holiday. Yeah, and he has very detailed notebooks, and they're all in Finland. And so Liana went back there. He actually had a couple of places he likes to go, but some of them were Finland, but are now Russia. And we wanted to go up here because not only is it an environment that's experienced rapid climate change, but you can separate out other drivers. There's not invasive species or urbanization or anything. So this is Frans Sillen's. This is Liana's. I think she's probably in the pretty similar spot. No one's built anything there. And Frans Sillen had these great notebooks. So it's sort of like a transact. He'd be like, I started at the church and I saw this shrub and I recorded the insects. And so we're able to re-walk where he walked. And again, this is another sort of fly-dominated system. So, but the main thing we found here is that there used to be a lot of moths. And again, Liana had much higher sampling intensity than Frans Sillen. He only went there for 2 weeks for his holiday. She was up there for a whole season. She even did 24-hour sampling. And of course, it's light for 24 hours up in the Arctic Circle. She still never found any moths. And she went for 2 years and never found any moths, which is quite depressing. Again, she found that the networks are becoming way more generalist and with the, yeah, anyway. I think, I've forgotten who the paper is by. There's this paper, it's from a while ago, where it's like this idea that in these really variable environments like high altitude and high elevation, that you'd expect plants to be more self-compatible because of this, to escape the variability. But even in the Arctic, we didn't find any plants that weren't pollinator dependent. Obviously, we didn't sample the whole plant community, part of the reason why is because the reindeers really liked our experiments and they kept eating the tags, which means you lose the plant. And even in this species over here, which is described in there's only one species that is maybe pollen limited, like it's all the sup and the open are not significantly different, is this species over here, and it is obligate moth pollinated. Well, it's described as. But it is still producing seed in the open, so something's visiting it, and we can't find the moths. But unfortunately, our sample size in the supplement on this one is only 2 because of the reindeers, which is probably my favorite way we've ever lost pollen limitation replicates. Usually it's crows. I think they're just curious. So probably one of the conclusions we had is these plants do seem to have generalist plant morphology, so maybe they're just interacting with a lot of things, and so they don't care, they're super generalist, so that's why they're not changing. And I really resisted this because I feel like orchids are overstudied. So in the pollen limitation data set, the orchids are up here. They dominate the data in this plant family. Like we have the plant families are well distributed, but this is almost all day orchids up there. And as you can see, they're super pollen limited. They are the most pollen limited plant family on a global scale. And that's because they're super generalist, super specialist. Orchids have high one-to-one pollinators, and that's because of the way they attract pollinators. So sexual deceptive species, you know, they pretend to look like a female of a particular species. They often release the pheromone of that female, and they trick insects into mating with them. There's also food deception, which isn't so specialized because they attract anything that wants nectar. They smell sweet. And you get some autofertile species, but it's not that common in orchids. They're mostly pollinator dependent. And I feel like orchids kind of deserve to be pollen limited because they often don't offer anything other than being pretty. The cool thing about orchids though, and the reason why we use them is because yes, they're specialists, so we're hoping we'll detect something. But they package their pollen in pollinera, pollen, and you can observe it whether or not they've been removed on a herbarium specimen. For a lot of species, there's a lot of species, they even protect them, and so it requires a trigger mechanism, and so it's either been triggered or it hasn't, and they're there. And the other thing that we did, so using herbarium specimens, we recorded pollinator removal through time, but we also looked for other evidence to support those observations, and so we collected information on ovary fertilization. And I think like this should be straight, but this orchid is very pregnant. My co-author gets really annoyed when I say that because plants don't get pregnant. I disagree, it looks very pregnant. We also recorded information on whether there was pollen on the stigmas or whether we saw insect parts on the stigmas that were insects that were relevant to their reproduction, or as best as you could. But the data I'm presenting is just on the polarium removal because it was 90% of the data, but they're all, we did analyze them together and they're super correlated. The R squared is basically 1, so they were super correlated. So we basically recorded female and male reproduction. They are completely correlated. So this is all data from insect visitations, it's just male reproduction. This is Bofen Song, he did his honors on this, and he collected data from Caladinia orchids preserved in the National Herbarium. This is about 11,000 individual plants that he collected information on. It's spread across the geographic range of Caladinia, the range is in red. They are the most endangered orchid genus in Australia, actually probably the most endangered plant genus in Australia. They're really endangered, and they're really handy because when they're in herbarium, they're quite beautiful in nature, they're like purpley colour. When they are preserved, the flowers, which are quite delicate, go see-through. So the polarium is both protected by the flower but also still visible. So we were able to do this without destroying the flowers, which is important. The data goes all the way back to, I think, 1825, but we cut off from 1925 because it's quite patchy before that. And one of the strengths of the data is a lot of people go back to the same places through time and collect the same species. So for example, Caladinia major, which is the main one of the main species here, we've got 800 herbarium sheets for that species through time. And each herbarium sheet, like Bofen has one down here, has usually got between 3 and 5 plants on it. So it's a lot of plants so it's well replicated in both space and time. We found approximately 60% decline in pollinator removal across all species. Down at the bottom here is the ones that are capable of self-fertilization. We didn't find anything, it's a flat line. And as we predicted, as I was talking about before, the more specialized plants where they have the one-to-one relationship, which is all the sexual deception here, it's huge decline, it's almost 90%. We really wanted to look at specific pollinator taxonomic groups, whether or not there was a difference in decline. You can really only do that with the food ones because the sexual deception of almost all thymine wasps, different species. So we wanted to do it with like broader taxonomic groups because some of these food deception are specialized on flies and bees and stuff. But the actual data on what visits them is extremely hard to find. We basically couldn't really find it. We did our best. We used this. There's a photographer, a famous photographer of orchids, and we went through his photo collection of orchids to try and see what was visiting them, and that's the best we had. And we didn't find anything, but I can't say confidently that it's across broad taxonomic groups, because of the way we were forced to collect the data. There's a lot of effort and then not much. The main driver is climate change. So these are partial effects plots. So there's a strong correlation with temperature anomaly, which is deviation from mean climate conditions. So this is like a 1.5 degree increase in average temperatures. We also looked at rainfall, found no effect. We looked at rainfall in many different ways, like deviation from condition from the location they were collected, deviation from 3 months before when flowering would have been initiated. We did it, analyzed it in 20 different ways and found nothing with rainfall. We use another composite of land use intensity called the Human Footprint Index, which is a measure of how disturbed a site is based on human influence. It includes things like roads and urban area, agriculture, everything. We found an overall effect that plants that are in more disturbed habitats have lower pollinator removal overall compared to more natural sites. But interesting, the more natural sites had the bigger effect of temperature anomaly, which probably reflects the fact that they have higher overall pollinator removal. I presented this data at Kew Gardens last year, and so they decided to try and replicate it in their collection using species from Central and South America and Africa, and they find really similar declines. It's about 70% decline in pollinator removal. So the final goal was to really look at whether or not plant reproduction is limited by pollen and if it's big enough to cause population extinctions. So this again is preliminary data. What we've done, we've used the global data set, which is the global pollen limitation data set, and compared it to compadre. And actually there's also some comadre data in here, which is the individual, both the individual and the two different, and IPM demographic data. These are often separate published studies, but one of the first things we did was remove anything that's like a different continent, like too far apart. So, as you can see, the vast majority of our data overlaps. About 2/3 of the data, actually it might even be higher, it might be about 75%, is the same study location, even if they're published in different locations. We did like a spatial match that we didn't allow any sites that are more than a certain distance away. So all of these sites, the demography is either done at the same site for the vast majority of the data or very close to. Species that are long-lived, it doesn't matter if they're pollen limited. I've talked about that before. So you would expect generation time and pollen limitation to be highly correlated, and they are. That's sort of the first stroke. The blue is the pollen limitation data, and the red is just the full data from the global demographic data set. And actually, I'm quite surprised that we have distribution across the whole distribution of life history strategies. I have to think about a better way to present this figure because it's really awful, I'm sorry. But this is a one-to-one line. But pollen limitation effect size, what we usually do is a odds ratio. Is it odds ratio? And so it's exponential, and then so along this axis is the percent of reproduction that would need to be removed for the population to start declining. And then this is the pollen limitation. So if it's in the grey area, the pollen limitation is high enough in that population, the population is not replacing itself at the time the experiment was done. So it's actually about 10% of the species, which is more than I imagined, but it might also be reflective of the reason why sometimes we do pollen limitation experiments. Maybe we're picking species that are a little bit at risk. So who are these species? These 4 species are listed on the IUCN as overharvested. So they're in really isolated populations, particularly these 2 species. I think they're described in the red list as like often in isolated fields or by themselves. So this could be mate limitation. So maybe the pollinators are still there, but the dispersal is too much. The other species in there, there's quite a few introduced species. Not necessarily invasive, but the species, the demography and the pollen limitation is outside of their native range. So this could maybe be mate limitation again, or the wrong pollinators, the pollinator mismatch. So I hope, I sort of like pollinator dependency in summary is like it's very widespread. Most plants need pollinators. I think we should definitely be concerned about pollinator decline. Pollen limitation is super common, but how much trouble a plant is in is very much related to their own traits, because they can, buffer it with their own ecology and life history. We can see massive changes in the plant-pollinator interactions and pretty limited effects on the reproduction or the function. But I think I hope I've shown that in specialist plants, it's actually, I was quite surprised like just how bad the orchids were. At first, I think when we first plotted it, we wondered if we'd done it wrong. And it was a bit reassuring when Kew Gardens did it too and found the same thing, but also really sad. So I guess I want to thank all my collaborators. I thanked all the students that I have included, all their photos, and everybody who gave me money. So thank you.
Thank you very much. Do you have any questions? Questions to ask?
Well, first of all, thank you for the amazing presentation with many results. I was kind of puzzled by the result where you showed you revisited the alpine flora from Australia, and you found less pollinators, but they have a wider diversity of pollinators per species. That was correct?
So it's like we find less. Actually, well, I think one of the big things, the flies are remaining. So we found the same number of flies, but we don't seem to be able to find a lot of bees. And it seems that the flies are just sort of filling in. So things that were previously seemed to be mostly pollinated by bees are now being visited by flies.
And do you think could be because there is a relax of competition, so then there is more resources available and more opportunistic species go in?
And another weird, I actually meant to mention this and I forgot to mention it, that it used to be very stable, the community. So in the past, because we've also done seasonal turnover and looked at how it changes through the season. It used to be, well, we've only got 1 year. In David and Nui's year, the community was quite consistent through the whole year. Now we have this really strong effect where the bees just drop off. and it's probably because of temperature. And then it's only flies. And so I think the flies are filling in and some bees aren't even showing up. But yeah, so there's now a really strong seasonal pattern in pollinator turnover, where there was never, far more.
Curious. And you expect plants to have a stronger pollen limitation or reduced seed set, or do you think they can compensate quantity by?
I think we did some work with some flies up there to look at how much pollen they're carrying, and they're actually, some of the flies up there carry quite a lot of pollen, and when we dissect their guts, it's full of pollen, so they really are visiting, and full of different species. They're not only visiting plants, but they're moving between plants. And presumably, if they're moving between plants of different species, they're also moving between plants of the same species. The pollen limitation data for the alpine sites are not completely finished, because I was actually having a meeting with my student about it this week. Some of the sups look pretty big actually, but it's also super variable. And so I was saying to him, oh, wouldn't it be nice if you just had a bit more data? Because I think some of these look like they probably are actually, because the effect size is quite big. It's just huge variances. And then he was like, oh, I did do it at the beginning and the end of the season, so I can increase all of these ends by 100%. And I was like, yeah, we should do that. And we should look at the beginning and the end, because you've got these turnover. Like for the species that you have to be, he didn't do it for all of them, but for some of them, he redid it because he lost a lot to crows. So some species he redid it again later in the season. So it's quite, we didn't know that the insects now turn over, and they do in both of the contemporary years. The pattern's there for both. Yeah, so we maybe for some of the plant species can improve those error bars, because some of the effect sizes are like, you know, and a difference of about 40%. So we're saying that that's actually, like, I know your ANOVAs are saying they're not pollinator, but I sort of think some of them are. So yeah.
Yeah, cool.
Yeah.
Is there other question?
Yeah, thank you for your talk. Really, many results together.
Sorry. As I was giving it, I was like, oh, actually, I said it out loud. It was an internal thought. I was like, oh, I think I still need just more connective tissue and a few less results. Even as I'm presenting it, I'm like, oh, this is maybe a few too many results.
But I was wondering for the other, I mean, you are always talking about pollen limitation, but you are not showing any effect on the thickness of the plant.
Yeah.
So I was wondering that first, and then if you have any of this reduction in the pollen load is really having a kind of effect, like a mismatch in the production with the plants. I mean,
Yeah.
Mostly for the consequences for the other side, because you are very focused on the pollinator side, but you are not reporting a lot from the plant side.
So there's a few things going on. So we've also collected up, I was talking about that earlier, that we've recorded pollen deposition on the insects' bodies in different locations. And so, because one of the things we were thinking is that there should be an increase in, you know, having the wrong pollen on the plants. We haven't got the stigmatic data, which is part of the reason why we haven't finished that. There's thousands of slides for somebody to look at, but we haven't done that bit, which is the other reason why I can't completely comment on the plant reproductive fitness, but we're getting there. But it's just like, from a plant's perspective, you know, like we talk about bees being the obligate pollinators and they carry all this pollen, but from a plant's perspective, to replace themselves, they need a single pollen grain and a single pollen tube to be fertilized and to produce a single seed, and they've replaced themselves. And so, you know, if a fly is carrying significantly lower pollen loads, you know, what really is sufficient for plant fitness? You know, and so I think from a lot of, not that we shouldn't care about, you know, bee declines, and there's certainly lots of things that are only pollinated by bees, but for a lot of things like, and then Liana's data is the same actually. Josh has found that the community is the same, and so is the abundances, more or less. But Lana actually had a huge increase in flies. So I think, not that I want a world full of flies, but for a lot of the plants, I think they are less fussy than I think we often think that they are, you know. And I think, but some of the things that the plants do are really cool as well, because we were collecting the insects, and one of my students swabbed different parts of the insect bodies. And so sometimes pollinators, he did it on flies. So even these flies that we think are super generalist because they visit everything, the pollen is only deposited on a really specific part of the body. So these flies can be carrying like 10 species, but one puts it right on the top of the head, and another one puts it on the neck, and another one puts it on the body, another one the legs. And so the plants, you're maybe a bit more clever than we give them credit for.
Maybe just so you could like counting seed set of the plants.
Yeah, we don't like to just count the seed set because reproduction can be limited by resources as well. And so the idea of the pollen limitation is like it isolates pollen receipt. It doesn't necessarily isolate the vector, which is why we also then have to do pollinator dependency, because if the vector is wind, it's not really related to pollinator services. Yeah, so that's the main reason that we don't just measure the reproduction.
How can I open it? Is that the question online? So could changes in plant phenology drive some of the changes in pollinator community species composition?
Yeah, so that's also some ongoing work. So my students are is looking at whether or not the changes in the turnover in the insect community that we're now observing that didn't previously happen, if it's better explained by the changes in the plant phenology or temperature or rainfall. For flies, it's like temperature. And for the bees, yes, it seems to be the phenology. He's not finished, it's the current chapter he's working on. But for the bees seem to be responding to phenological shifts, and the phenological shifts seem to be responding to changes in water availability from about 3 months before or so. So it's snow melt, yeah. So they've got completely different drivers, which is why one is doing quite well and the other is not so much.
Was there a question? I'm checking online as well. No? If not, thank you very much for your talk, and thanks you to everyone for coming. And we see you, I think, in 2 weeks, because next week is holiday, no? So next seminar will be in 2 weeks. So thank you.
Thank you.